it is of tremendous importance that we prevent something like that from happening again, and for that we need to know how it happened. A lot of framing in both directions. This is what I think is the important evidence. This is what I want to focus on today. Look at how synthetic viruses are made. Severe safety concerns. So the most similar viruses were found very far away from Wuhan. If you look at the precise figures from their proposal, they mentioned the sequence of the receptor binding domain they want to modify. SARS-CoV-2 is really unique with having that re-enclaved site. A guy in Spain programmed AI to search all the huge DNA databases for SARS-CoV-2-like sequences, and he found a 100% spike identical match. And when we looked at the frequency of mutations in these stitching sites, what we assumed is to find a 3% mutation rate, but what we found is 22%. The chance that this is natural is far below 1 in a million.
Wir müssen wissen, wir werden wissen. Let's try to talk about science. My name is Henrik Klages. I'm the guy who had the idea for TNG, and I'm also responsible for inviting Valentin Bruttel today. So if you don't like it, you can go to me and tell me that I made a mistake. I don't think so, but I'm willing to listen. David Hilbert, the famous mathematician, said, we need to know and we will know. Wir müssen wissen, wir werden wissen. And so I think it's important to look at potential facts or real facts and how they could be structured, how you could evaluate them, and maybe how the puzzle that is being there will be assembled. And I should say that you were very early with your papers. I read it. We were very early with the COVID response. We had a written emergency plan on February 9th, 2020, when I think most of the companies or governments didn't know what was going to happen, but TNG has advanced feelers or whatever you want to call it. And what you did, for example, was analyzing and saying the genome of this virus has interesting sections. It has a certain number of sections, namely 6, and the glue that was used to stitch them together is a particular glue, a particular restriction enzyme. And you have to know there are like 1,000 restriction enzymes that you can order. Yeah, and later, a lot later than after this paper was published, an American research grant proposal was leaked. That was a proposal to DARPA, the Defense Advanced Research Projects Agency. DARPA very cleverly said, way too dangerous, we're not going to fund it. It contained essentially a blueprint for this virus and was very interesting to see. It said that it's 6 parts, and it was a research grant proposal, so you had to have a long list of where you need your money for. And one of the items in the long list of what you need your money for was a particular restriction enzyme, which was the restriction enzyme that you predicted. And I thought this is a rather interesting coincidence. And therefore, we are very curious to see what you will think and say. And I should say, why now? A, because wir müssen wissen, wir werden wissen. And also, we're working in artificial intelligence. The artificial intelligence nowadays, we are not bad at that technology. It's rapidly getting stronger. If you talk to the large model developers, they say half our pipeline is already completely automated. The stronger model is iterating faster. It's getting better faster. It's like a snowball. Those words are very up to date. So that means even though the artificial intelligence technology has that large potential for good, it also has risks. The artificial intelligence is currently in a gain-of-function phase. It's rapidly gaining functions, like you learned from Outropic's Mythos. What functions will it gain over the next years? Will we be able to control them? What decisions will the people make who are in similar positions as biologists probably have been a couple years ago? And maybe we can learn something for the next time. So please blame me later and give an ear to Dr. Valentin Bruttel before I hand also to Dr. Stapor to give us a few more insights. Danke dir.
Thank you. Especially some introductory words. So hello, of course, and welcome everybody to our 3rd session of the day. Welcome to the on-site guests, which are very numerous, and also the remote guests. And with that being said, it's my great pleasure to introduce Dr. Valentin Bruttel. He studied biomedicine and molecular medicine in the beautiful city of Würzburg and earned his doctorate in tumor immunology. Today, he's Chief Scientific Officer at Toleris Biotherapeutics. working as a bioengineer and as an immunologist. On the private side, he's been very engaged in scientifically investigating the origin of the SARS-CoV-2 virus. And he's also on the advisory board of BiosafetyNow, a non-profit organization advocating for more lab safety. And this organization also supported the congressional inquiry in the United States. And since this promises to be a very exciting, very suspenseful talk, I don't want to keep you waiting any longer and hand over to Valentin. Stage is yours.
Welcome. You can hear me? Perfect. All right. Thanks a lot for the great introduction. I can skip almost my entire introduction slide on my background. So what we do as my day job is develop biotherapeutics for autoimmune diseases. We discovered a mechanism in pregnancy where the embryo teaches the maternal immune system to tolerate it selectively without shutting down the entire immune system. We want to adapt that to autoimmune diseases and actually also to long COVID and ME/CFS. I'm a board member of BiosafetyNow, so very interested in keeping labs safe and preventing pandemics, but I have no financial conflicts of interest, no virology projects or collaborations or anything like that. Very important, this is my private opinion, my personal assessment, not representing any of my employers, and this was investigated basically in my free time over the last 3 years. So why does the origin of SARS-CoV-2 matter? If we compare it to 2 world wars, actually we can see that it sits with regards to the cost that this pandemic caused right in the middle between World War I and II. And it also caused more deaths than World War I, according to a recent World Health Organization estimate. I don't want to go into details of how much of that was directly virus associated or caused by other problems caused by the pandemic, but it is of tremendous importance that we prevent something like that from happening again, and for that we need to know how it happened. This was also already taken ahead of me. Of course, new technologies bring new risks, which could be difficult to control, and what we are currently told about the origin is very, very diverse. So if you look, for example, at the European Union page, any manipulation of this virus or artificial creation is still put in the context of conspiracy theories. They say that no one is responsible. If we look at, for example, the whitehouse.gov page, then this is called a lab leak. If we ask virologists very early on, they said that they strongly condemn conspiracy theories suggesting that this has a non-natural origin. And now very recent, we have the United States Attorney's Office starting the first case against an employee of a funding agency who is actually charged with conspiracy against the United States. So it's a lot of framing in both directions. And unfortunately, in my view, very, very little evidence. So this is what I want to focus on today, just what I think is the important evidence without that many judgments. So let's just put ourselves in this situation. Imagine we have an airborne pathogen that kills many near a large biosafety lab. We have the Communist Party that blames contaminated meat. We have livestock that is killed and Western experts that don't get access to the lab, but they still confirm a natural origin. And everyone assumes that this is correct until something happens. And in this case, it was 13 years later, Boris Yeltsin admitted that this was a lab origin, which is actually the Sverdlovsk anthrax leak in 1979, which in my view has some similarities with the COVID origin. And what we can learn from that is that governments are not necessarily always completely honest. We've also seen that with the Iraq weapons of mass destruction claims, which were not true. Some experts can be sloppy or conflicted, right? The literature cannot always be trusted, but the evidence actually was very, very clear. Back then, all the cases clustered downwind of that facility where they had changed the filter and then had to stop the work and put up a note, which wasn't seen by the team next day. So they accidentally released these spores of anthrax. Then when you look at the patients, the lungs were affected, not the intestines, which speaks for an inhaled pathogen, not something that you eat. And if you look at the strain that caused the pandemic, was highly related to a typical lab strain. So the evidence can be very clear, but the conclusions can be quite divergent. So the question is, can we trust the experts? I think it's a bit difficult when there are conflicts of interest at play. For example, in the child abuse scandal in the Catholic Church, there are probably thousands of priests or bishops that knew that something was going on. Not one of them really spoke up and alarmed the public. VW cheat devices, a little bit similar. First evidence of cheating was from the 1990s, and almost to the last day, they kind of denied that and said this was like technical glitches in the measurements. So I think we should trust the facts. But to assess those and to assess the conflicts, it's interesting to go a little bit back and look at how synthetic viruses are made. This technology was pioneered in the year 2000 by Ralph Baric in the United States, who produced the first SARS-similar genetic virus. And this is done by putting this huge genome of 30,000 letters, 30,000 nucleotides, into smaller segments that can be easily handled and produced. Here they use these junctions, and to produce these junctions, they use restriction enzymes that act like scissors. They have recognition sites at which they specifically cut, so in this case the GCC and then GGC sequence, and they use some of those that produce different sticky ends. So these sticky ends in the DNA are single-strand overhangs, kind of stick together, and then you can glue them together with an enzyme called ligase. And because they are all different at the different junctions, you can have the entire virus reassembled in the correct order. So this is basically the breakthrough technology. And these restriction enzymes, most of them produce always the same sticky ends, which is not very good. You end up with segments that could form circles or arranged in the wrong order. But if you use enzymes like these, you get always different sticky ends and you get the correct sequence. Now, how do you introduce these restriction sites is by silent mutations. So there are two different types of mutations in biology. Some affect only the DNA or RNA code, and some also change the protein sequence. And you don't want to change the protein sequence, otherwise all your colleagues will say, oh, this virus is not functional because you put in a new protein. But with these silent mutations, you can introduce or remove these restriction sites without changing the function of the virus. And then what they did is they amplified these individual segments first, then digested them via these gene scissors, and then assembled everything into one big, large DNA construct called a bacterial artificial chromosome. And this can then be put into a cell that is highly susceptible, that doesn't have important defense mechanisms against this. And if you put this DNA segment into the cell does everything else. It transcribes to RNA, the RNA is translated to proteins, and then the proteins are assembled with the DNA, or the RNA in this case, and the membrane of the cell into an infectious virus. So it's a very simple technology. Just from code, you get something that can be lethal. And the technology is now so advanced that such a virus can be built for like material cost of $5,000 within 1 week. If you compare that to nuclear weapons, which are also very risky and highly regulated, it's probably 1,000 times more expensive, or at least, I would say, I don't know. And it requires significantly more money. And this is how I think this technology should be strictly regulated. Then later, other virologists are still breeding viruses. So these are not completely assembled, they're just adapted to humans. And these were referred to as nightmare or killer viruses. These are bird flu viruses that are 100 times more lethal than SARS-CoV-2. And they adapted them in a way so that they could spread via the air from human to humans. So highly risky experiments, which led to what I often refer to as the war within virology about what can be done, what should be done, which is probably a debate you're familiar with in the context of AI. So on the one hand, we had the Cambridge Working Group. They asked that we should pause the creation of these potential pandemic pathogens until we can completely assess the risk. And then on the other side, we had a group called Scientists for Science. These two gentlemen, Fouchier and Kawaoka, were the ones who built these nightmare viruses. And then Christian Drosten was also involved here early on. And they said that we should focus on lab safety. What they say, they were opposed to limiting the types of experiments. And then there was, in my view, lobbyism. So, for example, in Germany, some virologists asked that we have to introduce risks. In the Frankfurter Allgemeine, in the United States, we had Anthony Fauci and Francis Collins, the heads of the NIAID and the NIH, so the equivalent of the DFG, with a combined budget of more than $40 billion per year. So almost every scientist is dependent on these people in the United States that said that this risk is worth taking. So these experiments are needed. There still was a pause on funding for these risky experiments put in place under Obama, but this led to what I think is a movement to China, where this was not really regulated. So Ralph Baric started collaborating with Xixing Li from the Wuhan Institute of Virology. They made the first viruses in China. This is the segment map down here. And then these stars actually indicate the individual silent mutations that are introduced to add or remove these restriction sites that you need for the assemble and to generate these DNA segments. Only a year later, the first purely Chinese viruses were made. They used now better restriction enzymes that produce longer sticky ends, so they work more reliably. And then again, almost only 1 year later, they made the first experiments where they put in receptors from other viruses into harmless bat coronaviruses, which were predicted or assumed to be better adapted to infect human cells. And then they tested the infection capacity in mice that have the human receptor. So these are transgenic mice. And at some time points, they had a 10,000-fold higher viral load by putting in these new viruses. So the debate in the United States changed. They said, oh, gain of function is only the case when you mutate viruses that already have caused pandemics or that can already infect humans. And these bad viruses are not gain of function experiments. In my view, this is clearly gain of function. And the cautious side kind of lost this debate in this case. The ban on the funding was lifted. And then also like Peter Daszak, who led this organization in New York, EcoHealth Alliance, that brought a lot of money to China, got his funding for gain of function research reinstated. In 2018, we had this DEFUSE draft that was already mentioned, an application for a grant submitted to DARPA. And this is very important. This was actually leaked later by a DARPA employee. All the scientists involved tried to cover this up. It involved, again, about scientists that we already know, Peter Daszak, Ralph Baric, and Xixing Li. And this was not funded, but very similar experiments were. And we now, since just last week, know that Baric also received funding from Europe, from the Wellcome Trust around that time. And he might get into legal trouble because he didn't declare that for whatever reason. But we don't know what was directly done in these other grants because we don't have the complete documentation, unfortunately. So I mostly refer to the DEFUSE grant, which in my view is the key piece of lab origin evidence. And I find it very questionable that all the applicants, all the institutions, all the universities involved did not come out and say, look, this was our plan, we never did it. Look at our lab books, nothing to hide, but try to cover this up. Of course, the initial publication said that this is a natural virus. For example, we have this most cited publication called The Proximal Origin of SARS-CoV-2. Their key arguments were that this is a new receptor binding domain. We've never seen this before, and a new virus. So they would have more likely used an already established viral backbone, which of course can be the case, but of course also every new genetic backbone is sometime published for the first time. And if you cause a pandemic before, you're not going to publish the virus 2 years later. So it's, in my view, very weak arguments. And now we know what they actually debated in their emails, which is quite interesting because the head author told here, Anthony Fauci, that he and his two co-authors all find this virus inconsistent with expectations of natural evolution. So they said this doesn't look naturally evolved. We know another of the authors says this doesn't really exclude an inadvertent release, and that there is a nightmare of circumstantial evidence that has to be assessed. And we also know that actually the funders, so Anthony Fauci and Farrar from the Wellcome Trust, had a big impact on this publication. They wanted changes and they put pressure on the journals that published this. So the emails tell us actually a lot. But let's focus more on the science-heavy publications that claim a natural origin. First one was this one called the Molecular Epidemiology. This focused on two lineages, lineage A and B, that were both found at the market in Wuhan. And of course, one point where they're correct is that a lot of these early samples cluster around the market. Many were found at the market. Big problem is these lineages differ only by 2 mutations. We know from that Diamond Princess cruise ship that up to 3 mutations can occur in a single patient. So this is not really something that came to humans with a lot of difference in time, and it's just impossible to say if a mutation evolves in an animal or in a human. These are not animal-specific mutations. So they could have jumped twice from a patient to different humans or twice from an animal to different humans. It's just impossible to know. So it's very questionable with regards to the conclusions. They also ignored some of the intermediate genomes. And we also know that lab leaks have actually leaked several lineages before. For example, in the Sverdlovsk anthrax leak, a huge amount of pathogen was released, and that infected many people. And whenever you have a huge amount, the risk that you have several different mutations is much higher. So in a human-to-human transmission, it's probably just one droplet with a few thousand viruses. Most likely, most of them are identical. If you leak a few liters of virus supernatant or something like that, of course, there can be many mutations in one lab leak. And then also very recently, there were severe coding errors in this paper. Then the next paper discussing the origin in Science was more focusing on the proximity of the samples to stalls that sold, for example, animals at the Wuhan market, and the distance to the famous biosafety level 4 lab in Wuhan, which is across the river in the very south of the city. The major problem here is that all the samples that were tested that we know of that came from animals were negative for the virus, and that we already had thousands of different cases in humans around that time, according to Peter M. Barak from Denmark, who was in the World Health Organization Commission, and that there was the sampling bias. So they specifically looked for patients that had exposure to the Wuhan market and then sequence them. So of course, this leads to finding more patients from the market. And then there was some issues with deleted data by the NIH. And another trick they did is they simply showed the animal stalls, but for example, didn't show the toilets, which is a very enclosed space, right? You had this very open hall where viral loads can completely be blown out by the wind. In a toilet, you have many people in a very enclosed space. You could have higher viral loads because of that. And there also was this gambling room that was poorly ventilated just on top of that. So it could have been all from humans. And they didn't show the biosafety level 2 labs, which is problematic because we know today that when they asked Xixing Li at which biosafety level they did the experiments, she said at level 2 and 3. So if you just refer to the biosafety level 4 lab, which is far away, it doesn't really help. If you add the biosafety level 2 labs to the map, you get some that are right in the center of this outbreak. Biosafety level 2 is comparable to a dentist's workplace. You have a lab coat, you have gloves, you have goggles, but you don't even have to wear these simple face masks that we had to wear when we had to shop at Aldi, for example, for a few months. Biosafety level 3 and 4 are a completely different story. You have this pressure suits. In biosafety level 4, the entire lab is enclosed in air-filtered enclosure. So doing these experiments of biosafety level 2 is very, very risky. There were other severe safety concerns, for example, failures in safety equipment. I can't go through all the details, but there were 6 of them that are mentioned in this report just within half a year before the outbreak. And actually it was a report done with 60 Minutes, a US TV show, where they visited the lab, invited by the virologists there. And if you zoom in, you can see that they kept their coffee mugs directly on their workbenches, which is an absolute no-go. There are bicycles in the lab. So it's really a nightmare. This was when they invited TV to the lab. I don't want to know how it looks like otherwise. So anyway, let's focus on the more robust origin evidence. So first of all, I think the outbreak location is important. Of course, this was very obvious from early on. So the most similar viruses called Reggie 13 and Banal 52 were found actually very far away from Wuhan, about 1,500 kilometers, in regions where you have these bats that harbor these viruses. So the key question is, how did these viruses get to Wuhan without leaving any trace? And if you assume that this could have been a lab origin, What I did here is just look for the number of citations of Chinese megacities combined with bat and coronavirus, and you can immediately see that Wuhan published way, way more about bat coronaviruses than any other city, especially if you normalize it to the population. So a lab origin you would assume to happen in Wuhan, a natural origin you would assume to happen in Laos or maybe Yunnan. Then again, if you look at the DEFUSE proposal, they described there in detail that they had more than 180 already sequenced coronaviruses in the lab and more than 10,000 bat samples, and that they had sampling sites in Yunnan and Laos, and that they shipped these viruses always to the Wuhan Institute of Virology. Another big issue was that they took their databases offline in the fall of 2019. They say this was due to hacking, that they had problems with hacking. I don't know why this was the case for 6 years on, but there were actually several different databases. This was in detail investigated by a group called Geographic, independent scientists from all over the world who did a lot of lab origin investigations. And this would just be the perfect explanation how these viruses got from Yunnan or Laos to Wuhan. If you had either an infected researcher that maybe didn't even notice the infection because it was very mild in many of us, right? Or you had a sealed sample put in a plastic box that is then just shipped. The other big thing that jumped out immediately was how well this virus was adapted to human spread from the get-go. Yeah. So the comparison here is, for example, the green line of SARS-1, where you needed 8 months to get to 8,000 patients, and then MERS is here. You needed 12 months to get to 100 patients compared to SARS-CoV-2, where you basically have almost a vertical line. And this is very unusual for a natural virus, which usually needs a lot of time to adapt to the human receptors. And during that time, you have very slow spread. But if you look at, for example, molecular analysis of the receptor binding domain of the spike protein, then this is predicted to bind perfectly to humans, much better than to, for example, cats or bats or dogs or pangolins, which is a bit suspicious. This should have been initially adapted for an intermediate species. And again, if you look at the DEFUSE proposal, what they say there is they want to basically model interactions between different bat coronavirus receptors and the human ACE2 gene. They also want to screen for spike proteins that can interact with an additional human receptor, which was also found to interact with SARS-CoV-2. And of course, if you model and screen spike proteins for perfect binding to humans, and then you leak something like that, it's no surprise that this spreads like wildfire. If you look at the precise figures from their proposal, they mention the sequence of the receptor binding domain they want to modify. And if you compare that part, the receptor binding domain, with the highly related Reggie 13 virus, you see this red area. There are about 20% mutations in that area, while the rest of the genome is almost completely white, 1 to 3% mutations, right? So it looks like a replaced site in at least one of these viruses. The other side that sticks out is this fusion cleavage site, which I'll talk about now. So this was in the media for a little bit. A fusion cleavage site is something that enables a virus to infect different cells in the body. The virus sticks with these spike proteins to the receptor, to ACE2, but this doesn't help yet. The virus also needs to put its genome into the cell, and for that it actually needs to cleave off part of that spike protein, which can be done, for example, via trypsin, which is a protease found in the intestines. This leads to diarrhea and not so dangerous infections because it's very difficult to spread among humans. But if you put in furin, for example, which is also expressed in the lungs, you can have much better spreading via the air from human to human. And ACE2 is also expressed, for example, in the brain, which can lead to much more severe outcomes. And actually, there was this kind of, I'd also almost say, trend in gain-of-function virology since 2006 that they put these fusion cleavage sites into all kinds of viruses. For example, here in SARS-1, then there's one example where they put this in a porcine epidemic diarrhea virus, and then one where they put it into a MERS virus. This was done by Barrick and Xixing Li. And then also into these bird flu viruses, which are extremely lethal. Just to give you an overview, the lethality of most of these viruses is between 20 and almost 100% in piglets. while SARS-CoV-2 is much, much less dangerous, below 1% case fatality rate. So these are really dangerous experiments that were mostly done in Europe and the United States. And this is just one example when you remove the furin cleavage site from SARS-CoV-2, for example, in hamsters, they don't get sick anymore. So it seems to be really a key to enable viruses to spread into other hosts and to infect highly vital organs. If you look at the furin cleavage sites where it sits in the SARS-CoV-2 virus, and you look at related viruses, all these are the closest relatives, you can immediately see that SARS-CoV-2 is really unique with having that furin cleavage site and looks like an inserted furin cleavage site, which can happen naturally. But again, if you ask what is proposed in DEFUSE, they have this graph where they suggest to put in furin cleavage sites into the S1, S2 border, which is exactly this location. So this R is already there. You need 2 R's for the furin cleavage site. So this is basically the only place you can put it in if you follow this proposal. Yeah. In a 30,000 RNA letter genome, it's exactly at the site that they propose to put them in. And then there's one more point. They said they want to use human identical furin cleavage sites, which is a bit weird because they're very similar. Actually, furin is in many hosts, and but if you look at the complete sequence of furin cleavage sites, it completely matches this human enzyme that was studied at Ralph Barrick's university. So it could be the template. Yeah. Then there's the issue of no intermediate hosts. We still haven't found any animals that are known to harbor this virus or have an ancestral virus. These were found very quickly in the examples of SARS and MERS. There were some articles that claimed raccoon dogs were likely intermediate hosts, but if you look at the DNA in detail, you see actually a negative correlation here. So the more raccoon dog DNA you have, the less SARS-CoV-2 RNA you have, which speaks against this host. Opposite case when you look at human. And then there was one paper that tested this. They put lots of viruses into the nose of the raccoon dogs, and they got a short infection, but the viral load was about 10,000-fold lower than in humans, which is just speaking against these being good intermediate hosts. And actually, there was an ancestor found in a cell line, the so-called Vero cell line that I mentioned already, in accidentally leaked contaminating reads that were released with another experiment. And some of these viruses here had exactly the sequence you would expect from ancestral virus, because they have this TTC, which is found in the natural viruses and predicted for the ancestor, and which is no longer found in these 2 lineages that were found at the market. So the virus samples could very likely be actually a sample from the most ancestral human genome. And when you again look at the DEFUSE proposal, they mentioned that they want to use these Vero cells and that they want to sequence their DNA at exactly the company that sequenced this DNA. We also know that I probably don't have time to go into this in detail, they already uploaded a part of a 99.5% protein identical virus in 2018 to NIH servers, which is why they also had to then later release the rest of the RITG13 genome, which is much more divergent. But most of these most related viruses were actually handled by the Wuhan Institute of Virology. And then there was an interesting experiment with artificial intelligence. A guy in Spain programmed AI to search all the huge DNA databases for SARS-CoV-2-like sequences, and he found a 100% spike identical match, but with a process called codon optimization. So the RNA sequence is very different, optimized for expression. And these samples were taken in 2019, when officially no one knew what the sequence of this virus was. And they were actually also containing a trimerization domain. So this is a domain you add to generate a vaccine that can work independent of a membrane. In the membrane, these spike proteins automatically trimerize. If you want to use this as a powder vaccine, you need these trimerization domains to have them in the correct conformation. Again, if you look at DEFUSE, they wanted to create codon-optimized spike trimers and use these gel or powder vaccines for bats. So this would exactly fit to their proposal. But to end on the evidence, this is actually my own work. We looked at these restriction enzymes, these gene scissors, together with Tony Van Dong and Alex Washburn. And to briefly recap, we looked at how these viruses were built before. It was mostly these 3 gene scissors that were used, and these BSA1, BCMB2 were used in 2017 by the Wuhan Institute of Virology. They often use these 5 to 8 segments the longest segment usually below 8 kb, and then the unique sticky end. So if you look at the upper example here is the natural MERS virus. You have just 2 restriction sites, huge fragments. If you look at the lab variant, you have these fairly even spaced segments. And the same is the case with this Wuhan Institute of Virology virus 1. You have very unusual spacing, and then with the lab virus below, you have this very homogeneous spacing. So you have this Goldilocks zone, so to say, of 5 to 8 fragments, with the longest fragment being only about like 30% of the viral genome in length. And then if you look at SARS-CoV-2 here again, compared to 10 other lab-made viruses, it sits right in that window. This is not yet the strongest evidence. We assume about 1% of natural viruses also have that pattern. But SARS-CoV-2 is a bit different because it has these 2 unique BSA1 sites that allow for efficient replacement of the receptor binding domain at the fusion cleavage site, which is exactly the experiments they wanted to do. And unfortunately, you can read a lot about our publication in almost every German newspaper. Most of it is very negative, but you will never find anyone mentioning our key evidence. So the key evidence is shown by the lowest p-value, which indicates how strong the evidence is, how likely it is that this is of natural origin. And when we looked at the frequency of mutations in these stitching sites, what we assumed is to find a 3% mutation rate, but what we found is 22%. The chance that this is natural, especially in comparison to the Reg 13 virus, is far below 1 in a million. And I'm super happy to discuss this with anyone. No virologist so far was willing to discuss this. And then, as we already heard before, 2 years after our preprint, which unfortunately was rejected by most journals, they didn't want to send it out for review. And then there was this draft of the DEFUSE proposal leaked, which mentioned, as we heard before, exactly these segments and ordering BSMB1. So now the odds are even lower than before. I just want to summarize really quickly. I have to come to an end. There are, in my view, circumstantial pieces of evidence and then molecular pieces of evidence. I wanted to ask how consistent they are with labs or people or experiments in the DEFUSE proposal. And I think that we went through most of them. There was this issue with lab safety. We know that the outbreak started near safety level 2 lab where they did these experiments. It was also surprising that they shut down the Wuhan Institute of Virology, was taken over by government officials, and virologists were no longer to communicate, which is a bit weird in an outbreak. That should have been the expertise. There were suspicious emails and dishonest authors went through that. The deleted databases, eyewitness statements, I didn't have time for that. And then the timing and the location. So these viruses circle for thousands of years. The bat caves are very far away. Sick researchers, according to the US intelligence. And then if you look at the molecular evidence, we know that both these receptors they wanted to use work for SARS-CoV-2. We know that they had these related viruses synthetically assembled. The Reg 13 sequence is very weird. They mentioned the fusion cleavage site, which is unique, the first time in this family of 1,500 viruses. They mention the Vero cell line to produce these, which is then sequenced at the company, Sangon. And the plasmids are something you would use to do these kind of experiments, characterize receptor binding with just the protein and not the exact virus for which you need these plasmids that have been leaked into these samples. And then, of course, the restriction site pattern and the mutation frequency are also very suspicious, and I just don't know any natural explanation for that. So due to that, my conclusion is that a lab origin is now proven beyond reasonable doubt. I'm very, very open to discussing this and to opposing evidence, which has been difficult. But I think that, I don't have time to go over the personal consequences, but I'd really like to thank the people who investigated all this. I want to thank my family who went through a lot in this time, and then my co-authors. And with that, I have to come to an end. I think that we should really stop enhancing human pathogens. Otherwise, human pathogens may end us. With that, I'd like to have some time for questions. And thank you very much for your time and attention.